Can everyone hear me? Um, well, I'd really like to thank my friend Surwat for inviting me to this, and Dr. Professor Singh for inviting me. It's a great honor to speak here, and it's fantastic. There's such a nice turnout. Um, what I'd like to talk about today, actually, the session is primarily on, I guess, machine learning. We're actually not going to talk about that. And this is my group here. And my laboratory actually focuses on really physics-based solutions. And today I want to really just talk about two of them. And I think most people here are familiar. If not, feel free to ask. So it's an MRI scanner. And how do we observe structural changes that we know are occurring in Alzheimer's with MRI? And then I want to talk about something that's very new in the lab that we've been working very, very hard on, which is how do we observe functional changes. We're not talking about MRI, we're talking about EEG. And this is a very tricky problem. How many people here do EEG, are familiar with EEG? Hands? Some. Okay. I'll talk a little bit about that. And really, the one thing I want you to take away from this talk is how critical physics and basic neuroscience research is into getting clinical solutions. Everything I will show you today, none of it exists on a scanner. And the data we get, but the processing doesn't. It's a really critical issue. So here's early imaging. This is the way it used to be done. It's a hand drawing by Da Vinci. Can anyone tell me what that is? Any guesses? That is the first MRI image. And we've made some progress since then. And once you have scanners, you bring in physics. And I assume you're familiar with this idea, and we'll talk about how to look more closely at that. So I want to focus on Alzheimer's disease, and in which there's obviously a whole lot of physiological changes going on. And there are essentially two hypotheses. There's sort of the amyloid plaque hypothesis, but then there's also the neurofibrillary tangles hypothesis. And that's what we're going to focus on. And it's sort of our belief that this is the Brock neurofibrillary tangle hypothesis in the staging system, which was originally that the disease starts in the transcendental rhinal cortex and then progressively goes up through the brain to the neocortical regions and the primary secondary areas. However, this was modified to start in the locus coeruleus. And the locus coeruleus is this strange little area in the brain, which is very small. And that's where the challenge comes in for imaging. And that's what I'd like to talk about first today. So the locus coeruleus is here. This is actually too big. It's in the brainstem and has projections throughout the brain. So if that's where Alzheimer's starts, the idea is how do we look at that? And so here is a bit of a stain. You can see it's actually quite small. This is a blown up region. And here's where it is in the brain here. But it's actually really small. So this is a huge imaging challenge for MRI. So the first challenge is how do we quantify the damage and the neural pathway disruption? What we really want to see is how does the disease progress and how do we quantify that? So this is what we call diffusion. How many people here are familiar with diffusion MRI? Some people perhaps? Okay, so water molecules are diffusing around in the brain and all the time as we speak. And it turns out that MRI is actually sensitive to that, which is one of the remarkable things about MRI. But diffusion itself is a really fascinating physics. People love this. So if you take a drop of ink and you put it on a tissue paper and a newspaper, you get different looking shapes, which is really interesting. And why is that? The reason you get that is that you're getting a macroscopic picture of something microscopic. Okay, so if you look at tissue paper, it's sort of randomly organized, whereas newspaper has these highly organized, which is interesting. So in the brain, you can sort of think of gray matter as sort of being disorganized. And what we call isotropic diffusion means sort of just, there's no directionality to it. And in aligned fibers, however, what you get is diffusion that diffuses preferentially along those fibers. It diffuses in the direction along the fibers. So you can go right to what sort of an analogy with the human brain, which is gray matter here is sort of like this, and the diffusion in 3D is isotropic. And in white matter, what you have is something that's anisotropic. So I won't go into the details, but a different MRI technique, which is able to actually measure these diffusion profiles here, is called diffusion MRI. So we quantify that in the old days, and actually the scanners still do just this. Measure it, and something is anisotropic if it's non-spherical. So this is completely isotropic. And then you just calculate, it's a very simple number. And this, the bigger that, the more stretched out and elongated that thing is, the more anisotropic. And as I showed you, that should be what represents tissues. So what's amazing is in the early days, actually currently still, believe it or not, you take this is a standard anatomical image. And if you actually just plot that anisotropy without injecting anything into the brain, you can actually show pictures of where the white matter is, which is actually really amazing that you can do that. And here you show these little diffusion profiles. These are actually the measurements. So you can see gray matter here is kind of isotropic, white matter is anisotropic. It's a little bit of a problem over here, and that's where we get to the interesting story. I'll get to that in a moment. If you just look at the way those things are pointing, ellipsoids, they're following the fibers. Remember, the diffusion goes along the fibers, and your eye is actually, your brain is doing a lot of cool stuff. One is it's showing you what the pathways are. And so we can do this on the computer, and we call this tractography. So diffusion MRI can do local measurements, and then it can create these interesting maps, which are reconstructing tracks and long-range connectivity. So these are what would be disrupted, you would think, in a disease like Alzheimer's. There's a problem, however, and this is actually where the physics comes in, because it's an incredibly hard problem. This is an anatomical image that you would normally get if you went to the doctor, and you can see there's plenty of white matter. And over here, you see it looks like there's dark spots, which would be indicative of a disease. And it's purely an artifact of the way the data is analyzed. Why is that? The reason is if diffusion is isotropic, what the standard model for, which is now just modeling for all of these out who do modeling, you fit this and you get something which is isotropic. If you have two fibers crossing and you have the wrong model, what you get is the same thing. So there's no anisotropy. And this is called the crossing fiber problem. And for those of you out there who are neurophysiologists and know a lot more than I do about this, you know that one fiber, two fiber, none of those are good models. The brain is incredibly complicated. So actually figuring out what is causing all this and separating those is very hard. And if you look here, now you can see what the problem is here's gray matter, and you see that it's, isotropic, white matter, and you see where the fibers are crossing here is a big mess. So you get averaged out to something spherical. So we invented a technique. I won't go into the details, but if you're interested, there's a paper on it. Gets around this problem. And I'm showing you here that you can separate out an arbitrary number of fibers crossing at an arbitrary number of angles. And so this was a long investigation into how to do this. And it's both a computational problem and a theoretical problem. And for those of you who are interested in inverse problems, which I'll talk about, this is an interesting one. But what that allows you to do here is this is a standard fractional anisotropy map, and then here's the result. And you can see that these are scaled the same, so it's much brighter. And we use something that's called the equilibrium probability, which you can think of as a measure of anisotropy and connectivity. And more important for this, what we're showing is this is what we're now allowed to reconstruct. which is full brain tractography. And you can see here that you have this really amazing branching structure. There's actually this lends itself to a mathematical form for any mathematicians in the audience? Is everyone just shy? Turns out the brain actually has a very simple mathematical structure, which is absolutely remarkable. Notice, in fact, that fibers don't really cross in the way people used to think. This is more like a tree, which is not actually that surprising given what most of nature looks sort of like that. Okay. Yeah, it's pretty amazing, right? Now, what can you do with this? People who do tractography, what do you do with the tracks? And that's actually it was a whole brain full of tracks. So if you look at this flight map of the US, imagine these are all tracks. And if you just count from this airport to this airport, and you just count up the number of tracks, and the more tracks, the thicker the line. It's just counting. That's all you're doing. Counting between, I don't know if you guys are familiar with the US, but these are hubs of the airports. So thicker lines. And so we came up with a new, which does this. And you do that on that data that I just showed. It's really quite a remarkable image. So that's actually an MRI reconstructed image that looks like anatomy. And so this is called track connectivity modes. And it's sort of like doing dissection non-invasively. And so you get this. So this gives you a really detailed view from the tracks. So now, but going back to our original question, so this is my all-star student, Serafina, who's finishing her PhD. And the question really is, can you do tractography from the locus coeruleus to the transcensory rhinal cortex? So if you're just looking at the, people who do diffusion often just look at the corpus callosum, that's easy. That's just big tracks. This is hard, and you saw how tiny that is. So the result of all this work is that he's going from the locus coeruleus, and here's those track connectivity modes. So these are a nice summary, and these actually are just objects. So you can actually calculate, you can use this as a region of interest to calculate something. But you can see how complicated this region is. So without our method, our view is you can't actually do this. And in fact, no one has actually, only one other group has actually tracked this region and got a different answer. It's fine. So this is the result. So those red ones are the primary tracks, and it's very repeatable over multiple subjects. This is a second set, which is sort of less probable. But this now allows us immediately to look at that real question, which is, can we look along that and see? So these have spatially distinct. You can look at the anisotropy along that track because you think in Alzheimer's that that the disease progresses along that, and that is what we're beginning to see. And in fact, these are just initial results, but this seems to the EP is, so you're looking along that region, and it looks like what you're getting, it correlates with biochemical measures of the disease. So we're super excited about this, but you can see it's the end of a long process of doing physics and computation before you can actually do something like this. Okay, so that's a summary of this. Okay, now I want to go to the second part, and I want to talk about biomarkers. And so here what we're looking at is, so dementia, this is AD, Alzheimer's, mild cognitive impairment. So mild cognitive impairment, that's really where the action is in some ways, is where do you begin to see someone has the disease? And even that definition, so we're not experts in Alzheimer's. Mark Bondi, who's a neuroscientist, that's his area. Even how you define MCI is very difficult. And one of the ways that we're hoping to define it is through neuroimaging, to really do early detection. So here's about as good as you can do with MRI, but we've been looking at EEG. Now, EEG this is a great physics problem because in our view, it has never been solved. How many people here, again, have done EEG? Some people familiar, a little? Okay. Mild hands, little. Okay. So this is the state of the art. This is actually from our collaborator. And the work we did is in autism. This is just, in fact, the paper was only just submitted. So we work on this for a while. But the main thing to keep in mind relative to Alzheimer's is that autism is a disease that, well, a condition really, where it's an attention deficit. So there's problems with attention. It's you can't switch attention, actually, which is very strange. But it turns out that a lot of the work on Alzheimer's is also doing attention experiments. And in fact, Sir Watt's an expert in that. So there's a mapping between what we actually want to look at. So this is a hat. So these are electrodes, and you detect the electrode. So the brain is generating its electrical fields. So when people who do functional MRI, one of the problems with that is that's a blood flow effect. It's very slow. It's not really measuring brain activity per se. This is. The problem is this is the state of the art of how you reconstruct it. So here all you're seeing is a change in what's called the alpha power. There's a certain frequency band in the brain in typically developing kids or normals. And there's no change here. Okay, and that's the best you can do. There's no spatial localization at all. And there's a reason for that, is that it's hard. So this is sort of my way I think about imaging, which is you have a theory and then you simulate it. You have some idea numerically, perhaps, and then you have an observation of what you expect that system to give you. Imaging is the inverse process where you collect the data, you reconstruct it, and then you match it with theory. So I like to think of this as sort of two parts. Now, why is MRI easy? Hopefully that's not offensive to anybody, but MRI is really unbelievable. So this is actually quantum mechanics in action. Okay, it's really extraordinary, and the machines are extraordinary. But the result of the theory, the quantum mechanical theory, is that at every location there's a simple wave. So you can think of just the entire data set of MRI as just a wave, and that frequency is just a location, and that's it. So the answer is that's actually what the scanner detects. And if you can look at those, it's basically a bunch of waves. And the reconstruction problem is you just go from wave space to position space, and there's a simple mathematical formula that is actually every numerical methods thing. You just pump that thing through. So the theory tells us what this should be. That tells us how to reconstruct the data. Okay. Why is EEG so hard? Well, the measurements are only made on the scalp. You're not getting signals you're getting signals that are propagating, but you're only measuring on the scalp. So how do we know where the brain signal is coming from? And in fact, what do the brain electrical fields look like? So are people familiar with, generally speaking, electricity in the brain? So you have neurons, you have basically a current that goes down here. There's details of that are well known. You get an action potential, so sodium, potassium changes. And you basically get a current that goes down here. Right, anyone here a physiologist? Neurophysiologist? Somebody. You have to, okay, someone is. You guys have seen this. And so what you get is neuronal spiking. So this is very familiar, neuronal spiking. You look, okay, people are familiar. I see some people shaking their heads that yes. Okay. The model for this is an analogy. So Hodgkin-Huxley won the Nobel Prize for this. It's a circuit model. I'm not trying to be facetious. The brain is not a circuit. And there's a danger in analogies. So the standard model is that you have functional connectivities linked to structural. If I have neuronal bundles connecting A and B, that's how you communicate. So in the old days, I don't think any of you people are old enough to know what a phone is. Like a landline means if I'm talking to you, it means that our houses are connected by wires that look like this. Okay, in reality, the brain is made of complicated, inhomogeneous, and anisotropic medium, right? I mean, it's an incredibly complicated tissue. So is there a better model? So what do I mean by that? Inhomogeneous, this is white matter, gray matter. And you already saw this. It's anisotropic, meaning it has directionality. And these are some methods that we use here to do this. So the correct question is, how do electromagnetic fields propagate through brain tissue? And I always feel compelled to say this. If you look up physics of the brain, just Google it, and that is not what they do. What they do is they take fMRI data, for example, and they say, oh, this is connected to this. Let's make a network model. You're already not talking physics. It's a network model, but network models are great, but it's not physics. So how do you, so this is James Clerk Maxwell, the general equations of electromagnetism. And if you just think for a second intuitively, you have currents and charges, and that's just electromagnetism. Okay, so it turns out, I'm going to skip out, this is skipping over a lot, but you can actually solve this problem. So you can look at the brain tissue, and you know from MRI, you can look at the different tissues, and they all have different electromagnetic properties, conductivity, permittivity, standard material stuff, but they're very highly structured. So you can actually solve this problem. And what happens is you find surprise number one, because it's inhomogeneous and anisotropic, fields propagate in the direction of the spatial variations of these. So what we call the gradients of inhomogeneity and anisotropy. So this is remarkable. Waves actually propagate out. So this is not something you will have seen anywhere because it was not discovered before. So waves actually propagate outward because there are gradients in their changes in anisotropy and inhomogeneity. They propagate perpendicular to neuronal pathways. So listen to what, they're not just along them, but perpendicular. So that was not something that was known. And so this brain electrical field is waves, which is also not the way people think about it. And so these waves have frequencies and they have decay rates, and those are dependent upon the cells. And actually from a practical clinical perspective, that's super critical, because what's happening when you have Alzheimer's, your tissue properties are changing. Those feed directly into how you expect the waves to change. So both the frequency and the decay rate depend on this. So what you get are waves that have very low amplitude, but critically long time scales relative to the spiking. So you have these low level waves that are in the brain doing something. So this is what we call the universal theory of brain waves. And the fact that they persist is what's called weakly evanescent. And so we call these weakly evanescent transverse cortical waves. And so we had to come up with an acronym. Let's say Highland cow from Scotland. If you've ever seen them, they're pretty cool. This changes everything. In the old days, you talked between houses because they're wires. This is hippocampus. You can imagine there's, obviously stuff going along the neural pathways. But nowadays, that's how we talk. So different regions of the brain can talk even if they're not connected. That is a completely different way to think about the way brain communicates. It also means that when people make network models of the brain, they're missing a lot. Okay. So what are the other consequences of this? So waves propagate across extracellular space, not necessarily along neural pathways, large range of spatial temporal scales. The wave patterns I'll talk about in a second, it's very complicated. And between spatial extended structures. So A and B can talk even if they're not neuronally connected. Okay. You can imagine how much of a problem we were given when you present this. So any of you people out there who are in the publishing game, you get a lot of resistance. That's okay. So functional connectivity no longer necessarily linked to structural connectivity. That is absolutely critical. So how do these waves propagate through tissue? Um, I don't know if people here are familiar with classic rock. So this is what's called a dispersion relation. So you're familiar with the way a prism works, right? Which you have white light, and really all that's happening here is that white light has multiple wavelengths, and then the glass separates them because the index of refraction varies with wavelength. So it's something like this. So different ways to propagate at different speeds, and you get multiple wavelengths. So you can think of something's happening in the brain where there are all these waves propagating all the way around. So what does that do? Does that create anything coherent? So here's one very interesting thing is you can actually just take the tissue properties, and remember we had omega and gamma. You can actually take real properties, and here you can show that the waves tend to dissipate on the inner cortex, but they're supported on the outer cortex. And this is just a straight calculation using tissue properties. It's amazing how coherent these waves are supported, for example, on the outer cortex. So this is all done with actual real data. Okay, so how do these waves propagate? So here's something pretty amazing. Imagine you have a spherical brain. So you take the cortex, you just make a sphere because you want to see what's the the idealized situation. So that's your cortex, and it's the same all the way around. So it's idealized. And you solve the equations. Watch what happens. And this was a total surprise. So you solve the equations for these waves, and we were starting with a wave packet. You get a stable wave loop. Completely unexpected. This is a highly nonlinear system, which are often strange. If you solve this using mapping to a real cortex, you get the same thing. So if you watch where that goes, you see that you get a stable wave loop in the brain. So that's remarkable. You're getting a stable structure that's persistent long past neuronal spiking. And that's surprise number two. You have a wave trajectory that has an emergent loop pattern. And in fact, people have measured these already. So people have measured stable cortical wave loops. But there's no theory for those. So we think that this is a theory for how that works. So people have measured these, and they're coherent over fairly large areas, but they can be localized as well. So this is our data, well, data from Stefan Bickel. This is intracranial recordings, which is in epilepsy patients. And so you can reconstruct the stable wave loops from that. It's very hard. Obviously, there's a million things we don't know, and one of the things we'd love to do is animal experiments. So animal experiments would be really a fantastic way to do it. Okay, so you have all these waves. Do they interact with each other? And these are just idealized wave patterns. So when waves interact, you get interesting interference patterns and cooperative and non-cooperative phenomena. So in fact, they do interact. And so this is surprise number three, which is that when you actually solve these nonlinear equations, it creates spiking. So it's not very intuitive, but it creates spiking behavior anywhere. So for example, in the extracellular space, you can get spiking. Now, people have measured this. So the epilepsy people measure spiking in the extracellular space. And we call this collective synchronous spiking. And the way I sort of like to think about it is all these waves adding up, sort of the reverse of the other way to think about dispersion. And here's a really interesting plot. These are all, everything here is constant except changing gamma, which is it's the decay rate, but it's related to physical properties. So notice some interesting things. Weakly nonlinear, you get something that looks sort of sinusoidal. As you get more and more strongly nonlinear, Notice that shape. And this all depends, again, I just want to emphasize that if you use average, if you just use average properties, these all go away, everything decays. So it's completely dependent on the brain being anisotropic and inhomogeneous. So here's something interesting. Notice the shape here. It looks exactly like the Hodgkin-Huxley model, which was, if you actually go back and look at your textbooks, there is, the Hodgkin-Huxley model does not create spiking. The only way that you do that, because there's no resonance in that equation. So what they do is they add a few terms, and then they do something called integrate and fire, and it's essentially ad hoc. So this actually comes straight out of the electromagnetic equations. It's a completely different source for this. Okay, there's a lot of implications of that. There's also something interesting is you notice at certain values you get no spiking. It just turns right off, and that's indicative of what we would call a critical system. So it's on the boundary between spiking and non-spiking. So there's been a lot of talk about, is the brain a critical system? This would seem to be indicative it is. Okay, great. So now we are heading back towards, we have a brainwave model, which is the sort of a, just like in MRI, we have a wave model that allowed us to reconstruct. This is what we need in order to actually reconstruct EEG data. So we want to do the inverse problem now using this theory. So you have this, which is complicated time series from different sources. This has been said to be several times, but I like this one the most, about reconstructing the spatial distribution of EEG system was simply, it can't be done. So I've had that said several times. And it cannot be done using the standard model, which is fixed dipoles in a static field. That's not at all what's going on. So that's true. If you try that, you can't reconstruct it. Before we do that, I just want to, there's another problem that's embedded here, which I think is very familiar to people, which is, let's forget about EEG for a moment. Let's just look at a resting state experiment. And here, this is fMRI, where this little square is 1 voxel. This is time. So you've collected this volume 100 times. And this is just this little square here. So you have these signals in space. How do you analyze that data? And that is a problem that is ubiquitous in science. Like anything, we study weather too. Radar data is the same problem. You have spatial temporal patterns that are complicated. How do you reconstruct those? And how do you even begin to think about this problem? One way is coherent patterns in space and time, and that's a very generic term for a reason. I don't know if anyone who's a baseball fan. I use the term space-time trajectory, and all I mean by that is this is the baseball in space as a function of time. So it's a fancy term for something that you're already familiar with. You guys familiar with Mickey Mantle, but he almost hit one out of Yankee Stadium. Americans like that kind of thing. And if you actually look at that, so the ball is cutting out a path in space and time. Okay, and if we just look at a 2D picture of that, so here you're looking at nearest neighbors here, and nearest neighbors actually provide a lot of information, right? So nearest neighbors, we actually use this in that picture of the diffusion of the brain. We actually use that to reconstruct it, because it turns out there's always this problem about how do you connect a local phenomena with a global phenomena. And if you have things that are nearest neighbors, then nearest neighbors have nearest neighbors have nearest neighbors. So what we do is we use that, and it's not just in space, but it's in time. And it turns out that when you put all that together, you have this grid in space and time. This is 2D. And this grid in space and time is a space-time volume where if you have the correlations and the neighbors have neighbors and neighbors, the number of possible configurations of the system is actually quite small. It's a remarkable idea. Sorry, I didn't, let's see if I can go back. It's always dangerous. There we go. So we call this the entropy field decomposition. So EFD, and it's really for people who are interested in processing. I'm happy to discuss this afterwards, but it's a very powerful technique, and we actually do use it for weather and other things like that. And really, the thing to remember is that in most physical systems, the brain's the best example. You don't have sinusoids. You have things that are weird spatial patterns, and they're overlapping in space and they're overlapping in time. And the standard method that people use is what's called independent components analysis, but that doesn't actually work because they're not independent components, in fact. So here's just an example. This is independent components, and this is the same data set using EFD. And you get these very well-defined regions. And there's a reason for that, which is that something like this, where you have sort of noise, where it's sort of uncorrelated, or it just does, it's not, it actually doesn't matter. The right way to think about it, since this is actually a probabilistic method, is the probability that that thing is actually a real signal is so low. So it's actually a formal way to look at that problem. So this is single subject data. Okay, so now we're in position to do what we want to do last, I think, right on time. So now we have the brain wave theory. So we have a model for the signal, and we have a way of looking at space-time data. So we want to put this all together, and we want to basically figure out what the electrical field is in the brain. So what we do, here's the method. You have anatomical MRI data that gives you the tissue properties. So it gives you the anisotropy and the inhomogeneity, and people have done measurements of, what's the permittivity and the conductivity, et cetera, different tissues. You have the data. So that's using, and of course, and it goes without saying that morphology is complex, and all this depends on the morphology, right? Because everything's folded up. And then you have the wet cow theory. And then this is the EFD. Gesture is just our method for combining different data types. We just use acronyms because we can't keep it straight unless we do. And then you can solve that. So here is an attention experiment. This is for autism. And it's just, the task is control, and here's a target. All they're asked to do is look for red numbers. The ones on the outside are just distractors. But again, think about this, and since we're actually focused on aging, really it's just an attention task. And looking at alpha waves. So alpha waves are interesting because alpha waves are, as my neuroscience calls them, the idling frequency. So when you're actually paying attention, they decrease. So actually in these experiments where this is a typically developing child who is asked to pay attention, and when they pay attention, their alpha decreases. So this is actually a big decrease in alpha power, but sometimes we use the word activation. And you see that in autism, they don't. They just cannot switch attention. And so this is our result. So this is Spectre, and this is a reconstruction of the normal. So this is spatially localized electrical field imaging. So it's not what people call source localization. This is actually everything I just described, which is taking the wet cow theory and actually inverting it in the same way that we invert MRI. And you can see that the main thing we're looking for, this is our first time we've done this, which was just showing differences. And you can see clear differences. There's 20 subjects in each. And what that is that's the ventral attention network, which is activated in typically developing, but not in autistic children. And then we map this to an atlas where you can look at different regions. This is more for autism people. But this is a predefined atlas where you just look at how many, what's the activation in each of those. So you can use your favorite atlas for that. But just applied this to Alzheimer's, which is, and this is our first results. So we're excited about it. So that's spatially localized. These are, this is a verbal learning and memory. These are standard tasks for looking at memory. And we see patterns which are consistent with what is known clinically. So this is really just the beginning, but this is all sort of the physics that goes up to this. And so we're actively now working with sort of any experiment you can imagine for Alzheimer's and other as well. I know that people here are interested in perhaps mood disorder. That's part of well-being, certainly when you're aging, even in normal. And so we have some initial data in, this is just showing consistency in a task for people with a major depressive disorder and not, so very consistent results. And I want to finish with one thing. I think I have just the right amount of time. So we do also look at weather, and this is just a pitch generally to people, which is it's really interesting to work in multiple fields because they inform one another. So what I'm going to show you in the brain actually came from this. So this is a famous tornado. This is Goshen County, Wyoming. I guess you guys don't get a lot of tornadoes here, but in the summer, that's what we do. And people don't really know how those form either. So, but one of the ways we want to look at the structure, so storms have like an anatomy. It's really spectacular. They have a real structure to them, a very dynamic structure. We use EFD to look at those because it's a highly nonlinear system. But one of the things that we decided to do is this is velocity vectors in the tornado, and this is the inflow region. So one of the ways we thought to look at this is if you just look at the velocity vectors and you just you seed points along this, and then you connect those regions up together. So you want to see a pattern. It's basically like tractography, right? Except it's in space and time, and it's a little more subtle, but this is what you get, which is really amazing. I mean, so that's actually from the processed data. It's showing you the major modes, and here it's all about what we call vorticity, which is the turning of velocity. So we use this and we call these space-time information trajectories because they actually really are, they're sort of an information theory criterion that is doing this. But this really, you can just visually see what's going on. So I'm going to end by saying, once we did that, we thought, well, what happens if you use this with the brain imaging data? Because it's just space-time data. Again, it's the same processing. And I never get tired of looking at this picture. So those look like, those are brain electrical activity paths. And those are remarkably consistent. And these are tracks. So you can do the same thing that you do with the diffusion tracts, which is you can calculate the connectivity between regions. And so these are tracks, and these are the connectivity modes, and also remarkably stable, which is actually surprising. So we think this is a good way to look at electrical. You're actually looking at networks in the brain. And so I'm just going to end by saying that, we think of imaging as really not just taking pictures, but really exploring data. And everything I've showed here is really just the beginning, and it needs experts like you guys to be using it and really just exploring all possibilities and trying to help interpret what we're looking at, experiments in all, mood disorders and Alzheimer's and other things. But really, this is where we need people in neuroscience and physiology and all that. Okay, that's it. Thank you very much for listening.